Hello everyone, I'm Belaid, PhD student at Université Bretagne Sud. And this is my joint work with professors Elsa Dupraz, Laura Condé-Canencia, and Dominique Lavenier on channel model with memory for DNA data storage in a context of nanopore sequencing. So nowadays, the global data sphere, which represent the amount of produced and stored data, going up exponentially. And even though if a conventional storage system can be used, it will become more and more difficult to use them due to their ecological impact and also to the growing demand on raw material to produce them. So all these issues pushes us to improve or find alternatives to such systems. In this optic, DNA can be a powerful alternative, especially in terms of an archival perspective, thanks to its density, which is 1 million times better than hard drive disk and 1,000 times better than SSD or tape systems. And furthermore, it has a bigger durability compared to other storage systems, and it will never be obsolete as long as human beings will exist. So as other data storage system, DNA data storage works in two steps. A writing step where the digital data is transformed into synthetic DNA through a synthesis process, and a reading step where the DNA is where the synthetic DNA is read using a sequencer to retrieve the original data. In our case, we use a nanopore sequencer, which use nanopore, where the DNA data strand will go through nucleotide by nucleotide, and a current signal will be generated for each group of K nucleotide called a camera, and this signal is finally converted into base A, C, G, and T through a base coding process. To be more specific, in our case, we use the MinION sequencer, which is produced by Oxford Nanopore Technologies. And the sequencer offers some advantages, such as its size, which is small, and it offers longer and rapid reads with a relatively low cost compared to other sequencers. However, it has a higher error rate, which is about 10%. So to correct these errors, some extra steps should be added. And we observe on this slide a classic coding scheme with a channel and with channel encoding and decoding step, where we use the non-binary LDPC code. We also consider a specific channel where a consensus step is added, and this consensus allows us to reconstruct a consensus sequence from a multiple set of sequences. To save time and money, we use the simulator to test and validate our codes and results. So after exploring the state of the art, we found these next channel models, which are the IAD channel for independent and identically distributed channel, and the deep simulator channel based on a deep learning approach. However, both models seems to be not enough accurate to approach the experimental channel, as we can see it on these map editions, where the axis represent the position of the error on the sequence, and where the coordinate represent the sequence number. So in the case of the IAD channel, it doesn't take into account a burst of errors, but also the red camera, which can introduce more or less errors. And for the case of the deep simulator, we observed from our experiment that it seems to introduce the same type of error at the same position each time, and it forms there's vertical continuous lines, which are not so continuous in the experimental channel. Also, the predominance of errors are substitution rather to be deletions as in the experimental channels. So we needed to develop an accurate channel model for code design and performance. In what follows, we use xi to denote the red bars at position I, And we use yi to denote the observed event at position I. And we also use camera I to denote the previous read k bases at position I. Note that this core symbol could represent either an insertion or deletion depending on its location if it's on the reference or in the query. So after designing multiple channel model to try to approach the experimental channel results, we finally used the memory channel model, which is based on Markov chain with a memory of order k. And this channel uses three main characteristics, which are probability to observe the event yi given the camera I and the previous event yi minus 1. So this first characteristic allows us to estimate the probability to observe an event yi, but also to take into account burst of errors through yi minus 1. The second characteristic is the probability to observe an insertion of length L given a camera I, and knowing that we are in the case of an insertion. And the final characteristic is the probability to substitute the last base of camera I but by the base B, knowing that we are in the case of a substitution. We also take into consideration some specific case as described on the paper as the first and the last location of the sequences, as we observed from our experiment that these locations are has higher rates than other locations on the sequence, and it seems to be this error seems to be unrelated to read base. So note that the previous described model has a training phase where the previous probabilities are estimated, and once the training is done, the channel model can be used to simulate the DNA data storage. And as it can get more difficult to visually compare between channels using the edit map showed previously, we used the Kullback labeler divergence to compare between multiple channels. And we also add the bad-read channel, which is another simulator used in bioinformatics, but this channel need to be turned. So we use two cases for bad read. One case where we fixed the identity to 90%, and another case where we use the default mode of bad read. We can see on this figure on the axis the memory of the length of the memory, which is related to the memory channel. So all other values will be constant. Oppositely to memory channel, where it will vary depending on the value of k. We can see from this figure that when k equals 6 or when it is greater than 6, the memory channel model is better than the other channel models, and also that we think that fixing that k equals 7 could be the maximum value to use in the memory channel model to avoid overtraining of the memory channel model. Some interesting fact about this memory channel model is its adaptability. As if technology evolved, we can retain the model using new sets of experimental data. Also, since our model taking into account base coding, the simulations are faster, and also its explainability, as opposed to a black box approach as with deep learning. All our probabilities are explicit and can then be used to improve source and channel coding. So as a reminder, this is the DNA data storage workflow we used with the purpose to correct remain errors after the consensus. And we assumed at the first time that only substitution will remain at this state, but from our experiment, we observed that errors of insertion and deletions could also remain after the consensus state. So one of the main parts of our decoding scheme is the consensus algorithm, or a trace reconstruction algorithm, where the where a consensus sequence is reconstructed from edited replicates of the same sequence. The second part of the decoding algorithm is the channel decoding with the non-binary LDPC code. This code a linear code based on parity check matrix, which is sparse, and where any code word X verifies this property. And also for consistency, we used a Galois field of order K of order 4 to be consistent with the DNA alphabet. We also used a BP decoder. However, this BP decoder is commonly used in transmission field, which is commonly used in transmission field, can only correct substitution errors. So to improve these BP decoder results, we use a heuristic to synchronize the read sequence and allow better decoding. So in the heuristic, we do a synchronization, which allows to transform insertion deletions into substitutions, and this synchronization works in 3 steps. With a first step where where the sequence C is segmented into multiple sequences of length ls, and after that we insert arbitrarily a base A at a first position of each segment, and we compute a score E related to the number of unsatisfied LDPC parity check equation. And finally, we definitely insert the base A at position at the segment SG, knowing that the score G will be the minimum score observed. Also note that if multiple deletion exists, so this algorithm is used in a greedy manner algorithm. So the decoding scheme will become this way, where the consensus sequence is tested, and if its length is less than what was expected, then we use synchronization. Otherwise, we directly use, if it's equal, we use the channel decoding directly, or if if its length is bigger than what was expected, or if the decoded word is incorrect, so we use a consensus. We try to reconstruct another consensus using another set of sequences. So we can see on this figure the probability to successfully reconstruct the sequence using the consensus alone, then the consensus with LDPC with and without synchronizations. So in the case of barcode 1 and barcode 3, so in the case of barcode 3, we can see that that the synchronization allows to improve the decoding results until 60%. In some cases, but in the case of barcodes 1, we can see that in 3 cases the results remain quite the same. This difference is due to the fact that in barcode 3 an homopolymer of length 6 exists, which is a repetition of the same base, and this repetition introduce deletion during the consensus step. So in the barcode 1 case, homopolymer less than 3, so the results are quite good. So to conclude, the proposed memory channel model allows to model more accurately the DNA data storage channel, and the proposed decoding scheme improve non-binary LDPC decoding without any extra redundancy, as it relies on the LDPC structure. And finally, we plan to improve the proposed channel model by using genomic data, which are more available than DNA data storage. And we also plan to study complexity of the decoding scheme and use other heuristic that has better complexity than the one that is used. Thank you.